Ketan, are you there?
Yes, sir.
Ma'am has joined. Aziana ma'am has joined.
Yes, sir.
Okay. Good afternoon, Dr. Aziana. Aziana ma'am, good afternoon. Paula ma'am, good afternoon.
Good afternoon.
I think you have a session after this, huh? So now we have a session on Dr. Aziana Azamimi Abdullah from Malaysia. After that, only your talk is at 5 o'clock. I thought I have given you the wrong schedule. That's why I am worried. Anyway, I think Aziana having some problem. She is going to join again. Thank you very much for joining. I think some network issue, Ketan, huh? Aziana ma'am, again, I think she will join again.
Yes, she has joined again.
Okay.
She has joined again.
Once she will join, just inform me. Huh?
She has joined already, sir.
And Dr.
Yes.
Aziana, am I audible? You just unmute yourself. Muted, I think.
Hello,
Dr. Aziana, yes.
can you hear me?
Yes, we can hear you. Good afternoon, ma'am.
Yeah, good afternoon.
Okay. So I thought I mailed you in morning. I thought you have forgotten. That's why again I just rang you now. So thank you very much for joining, and we are waiting to listen about
So
that.
can we start?
Yes, Ketan, you can introduce Jana ma'am, then you can start. Yes.
Good afternoon, everyone. The third session is by Dr. Aziana Azami Abdullah from the University Malaysia Perlis from Malaysia. The topic for the third session is deep learning in biomedical applications. Miss Dr. Azian Azami Abdullah, please welcome us, a senior lecturer at University Malaysia Perlis, Malaysia for over 6 years. She obtained her PhD from Narla Institute of Science and Technology, Japan, and her bachelor's and master's of engineering in electrical and electronics engineering from the University of Tokushima. She has a history of working in big data analytics, AI, LaTeX, MATLAB, machine learning, Python, R, and computer vision. Let's join an informative session with her about deep learning in biomedical applications. Over to you, ma'am.
Welcome, ma'am.
Okay. Yeah. Okay, thank you for your kind introduction. So, can I share my screen?
Yes, ma'am. You can.
Yes, ma'am. Yes, ma'am.
Screen. Can you see the slide?
Yes,
Yes,
ma'am.
ma'am.
Make it full screen.
Okay. Full screen. Okay.
Yes, ma'am. It's okay now.
Okay. Okay, good evening, everyone. Thank you, Prof. Satya and the chairman for introducing me to this workshop. Okay, my name is Azian Azamimi Abdullah. I'm a senior lecturer at Faculty of Electronic Engineering and Technology, Universiti Malaysia, Perlis. Okay, first of all, I would like to introduce about my university. Maybe I'm the only Malaysian speaker for this workshop, so maybe this is your first time knowing about this university. So Universiti Malaysia Perlis is located at Perlis State in Malaysia. Perlis is the smallest state and located in the north part of Malaysia, very close to Thailand, border to Thailand. Okay, so I'm working at the Faculty of Electronic Engineering and Technology, Universiti Malaysia, Perlis. So in Perlis, okay, there are 3 main campuses that scattered around Perlis State. So the main campus is Pauh Putra campus. So this is my current location now, okay, Pauh Putra campus. So mainly for engineering, technology, and diploma program. Another campus is at Kanga. Kanga is the state, the center of Perlis, the Kanga City campus for Applied Sciences and Humanity program. And another one located at Universiti Alam campus Sungai Cucur. This one very near to Thailand, this one. So Universiti Malaysia Perlis has 3 campuses scattered in Perlis State. Okay, so this is some of the photos of Unimap, okay, my university. So this is the library, and Unimap located at the north, the hill, okay, on top of the hill. And this is the main entrance or gate. And this is our sports center or stadium. So this is, even though Malaysia is a hot season, okay, there is no 4 season in Malaysia, but we have spring season. We call it a spring season because usually in March there will be a tokoma season, this flower, okay, that blooming very beautifully every year in March. Okay, so in our faculty, okay, we offer diploma program, degree program, and also master and PhD, Doctor of Philosophy. So for diploma, there are diploma in computer engineering, and also diploma in electronic engineering. And for degree, the bachelor degree, we have 8 program, and I belong to this program, Bachelor of Biomedical Electronic Engineering. So in this program, we offer not only bachelor, but we also offer Master of Science and also Doctor of Philosophy. So if you are interested to continue your master or PhD, your postgraduate study, you can come to Universiti Malaysia Perlis, because we are among the cheapest, the tuition fee, the cheapest in Malaysia. Okay. So this is our Faculty of Electronic Engineering. So this is the pink building, mainly for admin office. And for the biomedical lecture and lab, usually we do at the blue building. So the facilities, we have 5 lab, okay, biomechanic lab, medical imaging and artificial organ lab, biomaterial lab, bioinstrumentation lab, and also research lab. Okay, so these are the equipment located in biomechanic lab. So those who are doing research on sports engineering or motion sensor or biomechanic, so they will do the experiment here at the biomechanic lab. And this one is medical imaging and artificial organ lab. So we have MRI here, and also some of ultrasound and other equipment. And this is biomaterial lab. So those who are doing research on biomaterial or biochemistry, so this is the wet lab conducted in this biomaterial lab. And this is bioinstrumentation lab. So those who are doing research on instrumentation or electronic, so basically the student doing their project here. And this is the biomedical research lab. So basically for final year project and also the integrated design project. Okay, so we have 6 research area: the medical instrumentation, and biomechanics and rehabilitation, biomaterials, signal processing, image processing, and also artificial intelligence. So I belong to this group for artificial intelligence research area. And our program organized the International Conference on Biomedical Engineering. So this ICOBI 2019 is the first, the conference that I collaborate with Prof. Satya, okay, the first first time I know him. So we are working on several projects. So until now, so hopefully we can continue this collaboration. So, and then for ICOBI 2019, first face-to-face conference. And then for ICOBI 2021, because the COVID comes in, occurred, so we had to do online, fully online conference. And recently for ICOBI 2023, which was held 4th to 5th September, this year. So we did the hybrid conference. So some of the participants attended physical participation, and also some of the participant attended online. So we call it as hybrid conference. So next conference will be ICOBI 2025. So if you are interested, you can submit your papers to our conference, because we publish in Scopus, Scopus Proceeding and also Scopus Journal. So now I would like to talk about my research area, which is the deep learning in biomedical application. So this is, I would like to introduce about my educational background. I did my bachelor's degree at Tokushima University, Japan, in electrical electronic. And then 2006, 2007, I worked as engineer at Toshiba. And then I continue my master's also at the same university, Tokushima University, Japan, in electrical and electronic. Then I became a lecturer from 2009 to 2013 at Universiti Malaysia Perlis. And 2013 to 2017, I went to Japan again to Nara Institute of Science and Technology to continue my PhD. And I obtained my PhD in 2007 in bioinformatics. So since April 2017, I work as senior lecturer at Universiti Malaysia Perlis, my current university now. And I also appointed as the program chairperson or program coordinator of Biomedical Electronic Engineering program. So my research area mainly in bioinformatics, artificial intelligence, AI, machine learning, and also big data. My current Scopus H index is 13. So for this workshop, Applied Python workshop, I would like to talk about my two projects. So the first project is the development of an improved model of COVID-19 mRNA vaccine degradation prediction system. So this is my PhD student work. So as we all know, COVID-19, known as SARS-CoV-2, is originated from Wuhan, China, which affected about more than 645 million and more 6.6 million death toll. So this one until last year, but I think recently the cases of COVID-19 has increased again. Even in Malaysia, there are a lot of new cases here. So the vaccines. So there are few different vaccine types. The first one is the inactivated virus vaccine, Sinovac. This is the commercial company that developed the inactivated virus vaccine. And then the live attenuated vaccine, the company is the NS1-RBD. And also the viral vector vaccines type, developed by Janssen and AstraZeneca. And then the subunit vaccine, Novavax. And the recent one is the nucleic acid vaccine, which called as DNA vaccine, the ZyCoV-D. And also another type of vaccine is the mRNA vaccine. Pfizer developed Pfizer, BioNTech, and Moderna. In Malaysia, I think most of the vaccine is from Sinovac, from China, and also from Pfizer. So the mRNA vaccine, known as a safe, high efficacy and also low cost to manufacture. But the problem with the mRNA vaccine is unstable. So what is the mRNA vaccine? mRNA that sends an instruction to the ribosome to produce S protein of SARS-CoV-2. Okay, so the problem faced by the mRNA vaccine is the degradation problems. Okay, degradation means not stable. Okay, not stable. So if the vaccine keep in cool chain, the life will be last around 900 days. Okay, if it kept in 2 units pKa, the life is around 10 days. Okay, for vaccination only 5 days. Okay, and if the vaccine is kept at 37 degrees Celsius, okay, the life will be around 5 days. Okay, so if in this hydrolysis magnesium 37 degrees Celsius environment, the life will be only 2 hours. Okay, and for in vitro transcription, the half-life will be last only 2 hours. So that means the vaccine will be fragile, okay, not stable, only can last for just few hours. Okay, so this instability will cause hundreds of thousands of lives were claimed were affected by this vaccine's instability. So what is the objective of our project? The first one is to devise and enrich architecture using hybrid modeling approach to give a better performance in predicting the degradation rate of mRNA vaccine. The second objective is to formulate a mechanism to utilize the hybrid model to improve the model of the mRNA vaccine degradation prediction. And the third one is to validate the performance of the proposed enhanced hybrid model with performance metrics. And number 4, to develop a COVID-19 mRNA vaccine degradation rate prediction system based on the enhanced models. Okay, so what is the scope of our project? Okay, the first one, the data set obtained from Kaggle will be implemented. I'm not sure whether you are familiar with Kaggle. Have you ever heard about Kaggle?
Yes, answer,
Okay,
student.
no. Okay, so I
Yes,
will
ma'am.
show you later the demonstration, maybe later. Okay, because we obtained the data set from Kaggle, this is very useful website. Okay, for the data scientists and also for machine learning engineers. And the data set will undergo pre-processing and feature engineering. Okay, proposed and enriched model will be developed using deep learning algorithm. So we propose a recurrent neural network, okay, known as there are two deep learning algorithms, which are the GRU, okay, the gated recurrent unit, and also the LSTM, okay, known as long short-term memory. And we develop the hybrid model with hybrid with linear models. Okay, so the enhanced hybrid model will be evaluated with a specified performance metric known as mean column-wise root mean square error, or the MCRMSE. And we also will develop an interactive prediction system for COVID-19 mRNA vaccine. And the Python is the main programming language in developing the model. Okay, so for the literature review, okay, we conducted a literature review for this topic. We found that not so many papers published yet about this study. The main reference is number 1, okay, published by Piaget Computer Science, okay, in 2021. The authors is Kite. So the title is Deep Sequence Modeling for Predicting COVID-19 mRNA Vaccine Degradation. So these authors, okay, proposed the LSTM model. They said that LSTM model has the best scoring score 0.125 for prediction when trained with the augmented data and cordon encoding method. The cordon encoding methods perform better than the base encoding method. The base encoding method is more recommended because base encoding has lesser tendency to overfitting than cordon encoding. The MCRMC difference for weighted average model with base encoding only 0.08, but for 0.016 for cordon encoding. Okay, so the remarks for this journal, maybe we could explore the effect of algorithm order in hybrid on model on results, and maybe we will study on test on 2 GRU hybrid with 1 LSTM, and also hybridization with 2 LSTM plus 1 GRU can be drafted. So another important paper is published by Journal of King's University, Computer and Information Sciences in 2021. Okay, the authors, the title of this journal is the iVaccine, Deep Prediction of COVID-19 mRNA Vaccine Degradation using deep learning. So these two papers use the same data set as ours. And for this second journal, the results is they found that the GCN GRU model has the best value of the MCR-MC score. The GCN GRU score 0.22614, which is quite higher than the first one. The first one is better using the LSTM. Okay, the private test score is 0.04, lesser than Kaggle competition winning models. The pre-trained GCN GRU has the best AUC score of 0.938. So they conclude that the GCN better than the convolutional neural network in predicting base-wise reactivity. So we found that this finding may explore results with bioinformatics encoding methods like dinucleotide, trinucleotide, codon, or also the tetranucleotide-based encodings. So authors only pretend the GCN GRU with non-embedding but fail to apply the technique on GCN CNN before drawing the conclusion. Could have detailed the justification on the hyperparameter for modeling. So this the topic can be explored later about the hyperparameter optimization. So from the journals, okay, we found that limited studies on predicting the degradation of the mRNA vaccine. So what we want to propose is to predict the COVID-19 mRNA vaccine degradation rate using the enriched hybrid deep learning algorithms. So the algorithm proposed for hybridization used to be from the same domain. So until now, from what we have read, so they only propose the hybrid technique using the same domain. For example, deep learning plus deep learning, or for example, the GRU hybrid with LSTM, or the GCN hybrid with GRU. So not many studies explore about the hybrid technique using the linear, the conventional machine learning hybrid with the deep learning. So what we want to propose is to hybridize algorithms from different domains. So from machine learning, okay, plus deep learning, for example, linear regression plus the GRU plus the LSTM. So we would like to propose this one. Okay, and then from the review, we found that there is not many studies conducted on the effects of the hybridization sequence. So we will consider the different sequences in hybridization algorithms in enhanced hybrid modeling. And then a no prediction system developed for vaccine degradation rate prediction. So what we want to propose is to design a prediction system to predict the COVID-19 vaccine degradation rate. Okay, are you with me now? Or is it okay? Can you understand?
Yes, reply participants.
Okay, okay, clear? All right. So
Yes,
I will
ma'am.
continue. Okay, so the methodology. Okay, first we extract the data. Okay, as I mentioned before, we use Kaggle to extract the data. Okay, then number two, of course, the data preprocessing and feature engineering. And then number three is the hybridization technique. So we would like to hybrid linear model with the gated recurrent unit GRU and also the long short-term memory or LSTM. Then we do the prediction with the enhanced hybrid model. Then we evaluate the performance, and finally we develop the prediction system using GUI. Okay, so okay, this is the flow chart, okay, of our project. Okay, first the data extraction, and then there are two data set. First with the BPS, and another data set is with training and also the augmented data. Okay, then we do data preprocessing and hyperparameter initialization, and then we select the algorithm. So for this case, we select for machine learning, we select the linear regression, and for the deep learning, we select the GRU and also the LSTM. Then we hybrid the GRU, LSTM, and linear regression, and then we train the model. Then we evaluate the performance, and finally we compare. And if the result is satisfying, then we will develop the prediction system. If the result is not satisfied, then we will go again to the hyperparameter initialization. Okay, so for first the data extraction. So we extract the data from Kaggle website. So the original the data is I think produced by the Stanford University. Okay, I will show you later the demonstration. So first the training data set. Okay, there are three data set. The without BPPS. Okay, the input, we have three inputs, sequence, structure, and also predicted loop type. And there are 5 outputs that we want to predict, which are the reactivity, the pH 10, the 50 degrees Celsius, and with magnesium, pH 10, and with magnesium 50 degrees Celsius. And for the augmented data set, and another is the with BPPS feature data. So there are 5 inputs, okay, 3 categorical and 5 numerical. So there are sequence, structure, predicted loop type, exponential weighted average, maximum normalized, and also position, average value, and summation. And of course, the 5 outputs is the same. First, the reactivity, the pH 10, the 50 degrees Celsius, this one without magnesium, and this one with magnesium. So for the data preprocessing, okay, first the label encoding, because for the RNA sequence, they use this character like AUGC. So we encoding using numbers, values. So 0, 1, 2, 3, 4, 5, 6 until 13. So we encoded the character. Okay, and then we do the data cleaning. Okay, so we omit the incomplete data. Okay, so this one without BPS, and this is the with BPS data. So, and then we develop the hybrid model, okay, the enhanced hybrid model. So we select 3. So the first domain is from machine learning, from linear regression, a linear variable model with input tensor and output tensor. And then we select the other 2 algorithms from 2 deep learning algorithm, which are the GRU and LSTM. The GRU is the compact with 2 gates, reset and update, and less computational time, while the LSTM complex with 3 gates. So input, output, and plus the forget gate, and it has better, it known as a better accuracy algorithm. Okay, so this is the structure, okay, sample architecture of our enhanced hybrid model with linear, GRU, and LSTM. So these are the example of 3-layer hybrid model. So this is the inputs. Okay, for 3-layer, the first layer we put linear regression model, and the second layer we put the GRU layer, and the third layer we put the LSTM. So this is the example of a 3-layer hybrid model, LGS. Okay, this is our short form, known as a linear, GRU, and LSTM. Okay, and for 4-layer hybrid model, so these are the example, the architecture example. So these are the inputs. Okay, the first layer is linear, second layer is LSTM, third layer GRU, and the fourth layer is LSTM. So this is the example of 4-layer hybrid model. So we put as label as LSGS, so known as linear, LSTM, GRU, and also LSTM. Okay, so we develop 44, okay, 44 models. So model number 1 until model 39 is our proposed model. So from model number 1 to model 21 is the 3-layer, okay, 3-layer hybrid model, okay, with different sequence. For example, this one, the first model, hybrid GGG. So the first layer is GRU, second layer GRU, and third layer GRU. Okay, for example, model number 21, hybrid SGL, mean the first layer is LSTM. Okay, G stand for GRU, second layer, and the third layer is linear regression. Okay, and then for the 4th-layer model is number 22 until model 39. So these are 4-layer hybrid model. Okay, for example, model 39, okay, hybrid GLSG means that the first layer is GRU, the second layer is linear, third layer is LSTM, and the fourth layer is GRU. And also, in order to compare with our proposed model with the one that proposed by other researcher, so we also implemented the same model as them, okay, as the authors, okay, number 44 until 40 until 44. So these models are proposed by KITE, okay, the first journals that we refer as our main reference. Okay, so for model number 40 to 44, they propose GRU hybrid 1, 2, 3. So just a few hybrid model and only GRU and LSTM. Okay, but they do not consider about a hybrid with linear model. So our idea is to propose 3-layer and 4-layer that hybrid with linear and also LSTM and GRU. Okay, so for performance evaluation, we use this performance matrix, the MCR-MSE, known as the mean column-wise root mean square error. Okay, so N is the number of predicted bases and NT is the number of targets. So the lower the value, the better the performance. So this is our indicator to indicate that which model give best prediction model, best prediction rate. Okay, so these are the results. So we developed 44 models. Okay, model number 1 until model 39 is our original idea. While model number 40 to 44 is the model that proposed by KITE. Okay, the main reference that we referred to. So we found that model 9 and model 27 give good prediction score. Okay, so this one is the without BPS data, and the second one is with BPPS. So if you see, if you observe that now model number 23, which is the hybrid model that consists of 4-layer, linear, LSTM, GRU, and LSTM. This is 4-layer model. Give a good result for epoch 200. Okay, the training score, the training RMSE error is 0.004, and for validation is 0.006. Okay, so this is very good results compared to the other researcher. Okay, so this is the lowest score that we obtain, and the time to complete the simulation is 0.77 hours. Okay, so the conclusion that we found from this proposed method, we found that we from our 3 objectives to devise and enrich architecture using a hybrid modeling approach to give a better performance in predicting the degradation rate of mRNA vaccine. So we have developed 44 hybrid models from 3 algorithms, which consist of linear, GRU, and LSTM. And the variation in result presented show the importance of hybridization sequences for enhanced hybrid model development. Okay, and the objective number 2 is to formulate a mechanism to utilize the hybrid model to improve the model of a mRNA vaccine degradation prediction. So the findings, we found that the hybrid model surpass regarding the time, complexity, and prediction performance, and hybrid models developed from different domains. So from machine learning plus the deep learning. And objective number 3 is to validate the performance of the proposed model with performance matrix. So we found that the prediction errors of all 44 hybrid models were evaluated with the MCR-MSE performance matrix. And the best model is model number 23, which is the 4-layer hybrid model, consists of the first layer linear, second layer LSTM, the third layer is GRU, and fourth layer is the LSTM. So the training loss is 0.0004, and validation loss is 0.0006, and the runtime is 0.774 hours. So this is the best result that we get so far. Okay, so before I discuss the second project, okay, let me show you some video for demonstration. But this one actually we're still working on the GUI for this hybrid deep learning model. So I just show you the previous GUI, okay, for the prediction system using the machine learning, okay, because this is our previous project. Okay, how to full screen, okay. Okay. Okay, so this is our source data. So previously we proposed the machine learning, LGBM, okay, linear regression and random forest. So this one without not deep learning. Okay, so this is our previous project, only machine learning. Okay, so the user can input the data, okay, the train data set, the test data set, and BPPS data set. Okay, so the user can explore the data. Okay, just click the button and all the visualization graph will come out. So this is the sample of final results from the GUI. Okay, so this is the prediction rate for 5 outputs. Okay, so the output will be in Excel file. Okay, so that's the first project. Okay, and then the second project, how to full screen this one? Full screen.
F5. F5.
F5. Okay, okay.
I think next go to that start screen. Here you will get somewhere the icon will be there. Slideshow. You can go to the
Wait,
slideshow here in menu.
Okay.
So there I think you'll find. Yeah.
Okay, the second project we working on is the development of prediction models to detect the presence of MGMT promoter methylation for prognosis of brain tumor. So for this workshop, I just explained 2 projects. Okay, the first one is the mRNA vaccine, and the second project is about the prognosis of brain tumor. So this one is a project for my final year student. Okay, project for final year student. Okay, so what is brain tumor? Okay, I think most of us know about brain tumor. Okay, it's a brain cancer actually. It's a 10 leading cause of death. Okay, for cases that below 20 years old, it's about 12.4%. For cases for patient for the age of 20 to 50 years old, about 28.2%. And the most cases is actually occurred for old people, like more than 50 years old, about 57.8%. So the majority of cases happen to the older people, that more than 50 years old. And 43.7% of the patients are males and 56.3% are female. So I think for brain tumor, the majority is female, and the age range is older than 50 years old, okay, larger than 50 years old. So for the diagnosis of brain tumor, there are 2 methods, non-invasive method and also invasive method. So for the non-invasive method of to diagnose brain tumor is medical imaging using the MRI, magnetic resonance imaging, the CT scan, and also PET. Okay, for invasive method is surgery. Okay, so the most common diagnosis method for the MRI is the for brain tumor is using the MRI. So these are the MRI sequences or the modalities. There are 4 modalities known as T1W. Okay, so this one is the short repetition time, TR, and short echo time, TE. Okay, cerebrospinal fluid is dark and the fat is bright. And another modality is T2W. So this one is the long repetition time and short echo compared to the T1W. The cerebrospinal fluid, the CSF is bright and the fat is light. So this is kind of opposite with T1W. And another modality is flare. It has long repetition time and short echo time with an inversion recovery pulse. The CSF is dark and the fat is light. And another one is T1WCE. So it has short repetition time and short echo time. The CSF is dark and fat is light gray. So now I want to introduce about the MGMT promoter methylation. So what is MGMT? MGMT is a DNA repair enzyme that may repair the damage induced by alkylating chemicals such as temozolomide, TMZ. MGMT is accepted as a biomarker in glioblastoma. Glioblastoma is a kind of a stage of the brain tumor. So MGMT gene promoter is either methylated or unmethylated. The treatment decision is based on the existence of the MGMT. So for example, for methylated MGMT, it will decrease the repair ability of damaged cells, so the TMZ alkylating treatment is effective. So if the patient has this MGMT gene, it means that the TMZ treatment is effective. If the patient does not have the MGMT, so unmethylated MGMT, so it means that it able to fix the damage produced by TMZ, making the TMZ treatment less effective. So the treatment decision is based on the existence of the MGMT gene. So for the problem statement, okay, if using the surgery method, so surgery is the only widely accepted approach to identify the MGMT promoter methylation status, which involve invasive technique and might lead to the serious consequences. So this is the main problem for for surgery, okay, for surgery method, because it is invasive and also might give a side effect or serious or consequences. So another is thermogenetic analysis, requires surgery to extract a tissue sample, and it can take several weeks to determine the genetic characterization of the tumor. So the problem faced by this method, first is the invasive, and the second, it might give side effect or serious consequences, and another is it will take long time to determine the genetic characterization of tumor. So the alternative method for diagnosis of brain tumor is to by using the medical imaging or MRI. But from the MRI, we would like to propose a method that can predict the presence of the MGMT gene, MGMT promoter methylation status. So the objective of this study is first to implement the data visualization techniques and exploratory data analysis EDA on the brain tumor data for better understanding of the data sets. Okay, number 2 is to implement deep learning algorithm to predict the MGMT status of the genetic subtype of glioblastoma through different sequences of the MRI, the magnetic resonance imaging MRI. And number 3 is to develop a graphical user interface GUI to help the users to identify the status of the MGMT promoter for brain tumor prognosis. Okay, so the scope of work. Okay, first we obtain the data set. So data set obtained from BRADS. Okay, BRADS is the brain imaging. Okay, from the brain imaging society. Okay, will be implemented. The second scope is EDA, the exploratory data analysis, and also data visualization. So data set will be explored and visualized for better understanding. And number 3 is image processing. The data set or image will undergo some pre-processing technique. And number 4, the deep learning algorithms. We will determine the suitable deep learning algorithm for prediction of the MGMT status. And finally, we develop the GUI, the graphical user interface, to make an interaction between users and the system. So this the research gap. Okay, we found that there are less references on the MGMT methylation status, methylation detection via using the MRI, and lack of studies regarding analyzing the sequences of MRI and comparing them, and the absence of user-friendly interface. So there is no system that has been developed to detect or predict the MGMT methylation status. So the methodology, first we will implement the data acquisition method, okay, from the BRACS data set, and then we implement the exploratory data analysis and data visualization to understand more about the data. Then we undergo the image preprocessing method. Then we implement the deep learning models, okay, to the 4 modalities of the MRI: FLAIR, T1W, T1WCE, T2W, and finally, we evaluate the models. If the result is satisfied, then we select the best model and we implement the GUI design. If the result is not satisfied, then we go back to the image processing and process the data, and we undergo the deep learning algorithms, okay, to predict the MGMT promoter methylation status. So for the data acquisition, okay, for training set, there are 582 samples, which consists of 280,000 images. Sorry, 347,280 images. And the train label consists of 2 source file. Okay, first is the CSV file and another is the DICOM. Okay, DICOM means the MRI images. Okay. And the testing set consists of 87 samples that have 65,851 images. So these are the examples of the MRI images for 4 modalities: the FLAIR, the T1W, T1WCE, T2W. Okay. And this is the example of the images that consist of the methylation, the MGMT methylation 1. And this is 0, means the images that do not have the MGMT gene. Okay. So the first step is to implement the EDA and data visualization. EDA and data visualization are crucial for understanding patterns and trends in a data set. They uncover hidden insights and relationships, guiding further analysis and decision making. Train and dataset will be discussed in this section. Okay, so this is the MGMT status of trained dataset. Okay, for from the training dataset of consists of 347,280 images. Okay. So from the dataset, training dataset, 47.5% is unmethylated. That means 276 samples have unmethylated MGMT promoter in their brain tumor. So this one, no. Do not have the MGMT promotion methylation status, unmethylated. Another 52.5%, about 306 samples, have methylated MGMT promoter in their brain tumor. Okay, so from the training set data analysis, okay, from EDA, so these are the 4 modalities. Okay, so this is the numbers of the files. Okay, consists of the FLAIR, T1W, T1WCE, and also T2W. Okay, so this is the testing set analysis. Okay. So for the second step is the image preprocessing. So first, the data acquisition. Second is the EDA or data visualization. Then the third step is the image preprocessing method. So why we implement the image preprocessing? Because first, to remove the black images. So allow the model to focus on key pattern and features. So the black images means we remove the black images of the MRI. And then step number 2, the value of interest lookup table application VOI LUT to increase the contrast and visibility of region of interest within the DICOM files. And number 3 is the image normalization, rescale the pixel value to fit within the range of 0 to 1. And then number 4 is the image type conversion from 16 to 8, smaller file size, has some original DICOM, image geometry to preserve crucial spatial information. And number 5 is to resize the image resizing to reduce computational and memory requirement. So this is the example of the pre-processed image. Okay, this one is the original image of DICOM, and this is the after we pre-process the image. So same as this one, this is the original DICOM image, and this is after the pre-processed image. So the comparison, okay, for the size, okay, original image is 0.5 MB. So after we pre-process the image, it's only 0.25. So it's easier. It requires less computational time, and it will be faster, okay, to process the image. And then the format, okay, from 16 to 8. So we reduce. So for the pre-processed data, okay, so the goal is to accurately classify the images into 2 categories, methylated and unmethylated for each sequence, for each modalities. The ratio for training and validation set is 80-20. Okay, 80% training, 20% testing. So the flare images, okay, the training is 42,883 and validation is 10,721. For T1W modalities, the training is 46,466 and validation is 11,617 images. And T1 WCE modalities, the training set is 56,528 images and the validation set is 14,133. And for the T2W, the training is 56,408 images and validation is 14,102. So there are a lot of images. So that's why deep learning is suitable to be implemented to process, to predict the methylation status. Okay, for the deep learning algorithm, okay, for this project, we use the convolutional neural network. Okay, the first project we use a recurrent neural network, GRU and LSTM. For this project, we use ResNet 50. Okay, deep residual convolutional neural network. So it's a part of CNN, and also EfficientNet V2. It's a compound convolutional neural network. So for the depth of ResNet 50 is 50 layers and EfficientNet V2 is up to 182 layers. And parameters, there are a lot of parameters, 23 million. And this one is about 4 million for EfficientNet V2. And the added layers, for example, the global average, pooling, dropout, dense, and same as the EfficientNet V2. So we select 2 deep learning algorithms and we compare which one give a good prediction rate for the methylation status for prognosis of brain tumor. So we utilize the ResNet 50 and EfficientNet V2. And we use sigmoid activation function, use the output layer for probability predictions. Okay. The loss function used is the binary cross entropy. And the MRI image slices treated as individual images. Okay, for the performance evaluation, we use the confusion matrix. Okay, maybe you are very familiar with the confusion matrix. So, true positive. Okay, true positive means that for methylated cases, the actual is positive and the predicted is also positive, means methylated. But for false negative means if the actual is positive, but we predict the negative and means unmethylated. Okay, so also same as false positive and true negative. Okay, for the performance metric, we use accuracy. See, accuracy is obtained by true negative plus true positive divided by all, true negative plus false negative plus true positive plus false positive. And we also use precision. Okay, precision is obtained by TP divided by TP plus true positive divide by true positive plus false positive. And for recall, true positive divide by true positive plus false negative, and F1 score, okay, using this equation, true precision times recall and divide by precision plus recall. Okay, so these are the model comparison. Okay, this is the results using two deep learning algorithm, ResNet 50 and also EfficientNet V2. So we implement ResNet 50 and EfficientNet V2 for every modalities. Okay, there are four modalities. So we implement ResNet 50 and EfficientNet V2 for all four modalities such as flare, T1W, T1WCE, and also T2W. So from the results, okay, from this table, okay, we compare in terms of accuracy, loss, precision, recall, and F1 score. So from our result, we can see that the T2W modalities, okay, outperform other sequences compared flare, T1W, T1WCE. So this highlighted color give the good results. So for example, the accuracy is 0.9453, means 94.53%, okay, for ResNet 50. So it obtained highest precision, recall, and F1 score around 95%. And the T2W image provide essential information concerning fluid accumulation, edema, and lesions, and effective utilization of unit traits and pattern. So compared to EfficientNet V2 and also ResNet 50, we found that ResNet 50 give good results for these two T2W modalities compared to other modalities. Okay, after we found that ResNet 50 give good result, then we develop the simple GUI, the graphical user interface, okay, like this one. So the user can upload the images, the DICOM format, submit, and then, okay, it will come out the original image and it will give the prediction, okay, the status of methylation, whether methylated or unmethylated. Okay, and then it also give a suggestion treatment. Okay, if methylated means the TMZ treatment is effective, but if the brain tumor MRI images, we predict that unmethylated means the TMZ treatment is not effective, so we have to find other treatment method. Okay, so there is no point if we just suggest to proceed with the TMZ treatment. Okay, so I will show you demonstration, just a simple demonstration after this. So the conclusion from the second project, so this study successfully achieved all set objectives. The first one is data visualization ADA, conducted data visualization and exploratory data analysis to gain insights into the brain tumor data set. And for the deep learning algorithm, we utilize the deep learning algorithms, the convolutional neural network such as ResNet 50 and also EfficientNet V2 with different MRI sequences, okay, to predict the MGMT status of glioblastoma. We found that the T2W modality consistently outperform other sequences in predicting the MGMT status. And we also developed the user-friendly graphical user interface GUI for the MGMT promoter status identification. Okay, so maybe I will show you some demonstration. This one. Okay, can you see now? Okay. Can you see the,
Yes, ma'am.
okay, so these are the examples. So first, submit. Okay, so this is the original image. And then our algorithm give the prediction status. So this one methylated, for example, and give the suggestion status. Okay, maybe it's too fast. For example, this one. So when we upload the image, for example, so our algorithm will predict the MGMT promoter methylation status. So for this one, it gives the methylated results. Okay, and if methylated means it also gives suggestion. It means that the temozolomide, the drug, the TMZ treatment is effective. So we can proceed with this treatment. But if unmethylated, I think the previous one is the example of unmethylated. Okay, this one methylated. And then if black image, then it gives you the result invalid. Okay, think not methylated. This one. Okay, this is the example of unmethylated. Okay, so it gives you the results of status unmethylated. Okay, too fast. Okay, for this one, when we upload the image, DICOM image, then our algorithm will give the status unmethylated. So if unmethylated means the TMZ treatment is ineffective. So that means we cannot proceed with the TMZ treatment. So the doctors need to suggest other treatment method. Okay, I think that's all for my presentation. Do you have any questions?
Yes, feel free to ask question.
Okay, I think
Okay.
that's all for my presentation.
Yes, somebody has asked in chat box, ma'am. Please tell me what are the softwares you use training and validating this data.
Hello?
In chat box, you can see somebody has asked question in chat box. You can take that question. Please tell
Okay.
me. Yeah.
Okay, please tell me what are the software you use in training and validating 50,000 images. Okay. For this second project, I think my student use the yeah GPU.
Some disturbances is there. Yeah. Continue.
Can you hear me?
Yes, yes.
For the brain tumor project, okay, the student use GPU, the personal laptop, but it consists of NVIDIA GPU, so it can process the images. Okay, if just CPU, I think the is not capable to process the images or to train to validate the 50,000 images. So we not use the cloud technologies.
They can go for this cloud pro, na? Google
Yeah,
Colab.
we use GPU hardware NVIDIA. So
Okay.
yeah. But if you have problem with the hardware, I think you can for example, using Kaggle, okay, like this one, you can use their cloud.
Yes.
Can you see this one?
Yes, yes.
Okay. So for this is the Kaggle website. Okay, you can go Kaggle. So this is the data set that we working on. Okay, so you can go to this and data. Okay, for the data is quite large. Okay, because it consists of 136 gigabyte, so very large. So if you have problem, if your computer does not support this very big data, because the data consists of the DICOM format and also the CSV, you can use the Kaggle notebook, this notebook. So for example, this one, okay. So quite slow. Okay, so you can go to the, this is data. Okay, data. Okay, you can download the data. Okay, if you have a good computer, a GPU, then you can download the data and you can process using your computer. But if you don't have that facilities, you can go to the code. Okay, there are a lot of data scientists give some example of the notebook, the coding. So you can go and you can try on your own. Okay, for example, this one I select for example, okay, this notebook. Okay, so this one, okay, it give you some of the coding and you can learn on your own. Okay, so for example, this one, and it gives some explanation so that we can learn. Okay, so this is the sample coding. It use torch. Okay, and okay, this is the results. Okay, load labels. Okay, and it give you the results like this. And okay, and the this person, sometimes it give you a description so that we can learn on our own. So that for example, load labels. Okay, and this is the result that he obtained. And then okay, he used the TensorFlow. Okay, like this, and give you the results. Okay, and this is the sample of the images. Okay, then training. So this one, it use the convolutional neural network, say CNN. Okay, then print the model. So give you the results and also the prediction result like this. So for example, this one show result of prediction. So this is the prediction result 1 and the actual is 1. So this is correct, true positive. This one, for example, 0 unmethylated, and the actual is also 0, mean unmethylated. So this is true positive, is a correct prediction. But for the third image, okay, the prediction give 1 means methylated, but the actual is 0 means unmethylated. So there are sometimes the deep learning cannot give you the 100% accuracy. But if the deep learning can give more than 90% accuracy, then it's considered good. Okay, so if you don't have the GPU, you can copy the code. Okay, like this. How to move this one? Okay, so for example, I just copy. Okay, this is my notebook. Okay, so you can use the cloud, the Kaggle notebook cloud, and you can run. Okay, try to copy first, then edit later. So for example, I run. Okay, so this using the Kaggle cloud service. So for this one, still working. Okay, so now it's still running. Okay, so for example, when you copy the code, okay, so it gives you like this and it gives you the results. Okay, so for example, this one, okay, so this is the MRI ID, okay, the image. So value 1 means methylated and value 0 means unmethylated. Okay, so this is the CSV file. Okay, so means that for the image 000, the status is methylated. This is the actual one. And then this is the training data set. So when we go for training the data set using deep learning algorithms, then it gives you the results either 0 and 1, 0 or 1. Okay, then we do the exploratory data analysis or the data visualization. So we found that for the training data set, okay, more than 300 samples is 1, give the status 1, okay, and this is the sample of 0, mean unmethylated. Okay, so still working because it consists of output is 6.1 megabyte. Okay, so for this one, save the image. Okay, so still processing. So at least it can save your computational capacity. So it's good that we can learn using this Kaggle website. So this notebook still running because the CPU is max already and the RAM is about this level. So it's still working. So you can try, okay, if you have time, you can go to this and try to register first. Okay, register first and then you try to run on the Kaggle notebook. So you can use their service, cloud service. Am I answering the questions?
Is it clear to you?
Yeah? Is it clear?
Yes, I think it's clear.
Yeah.
So another
So
question.
this one,
is there.
another questions?
Yes. You can check in comment box.
Chat box.
Chat box.
Okay. Okay, while waiting for the results to come out. Yeah, GPU in Kaggle is free for specific, yeah. So I think it's limited. If we use their free services means it's quite limited. So of course, the other approach, maybe you can use uh the Google Colab. Okay, Google Colab. So you can use either Kaggle notebook, okay, this one cloud service, or the Google Colab, but this one also limited. If you want to have more memory, so you have to purchase, you have to pay. And if you have, I think it's good approach is if you can find a good GPU, so you have to buy a very good computer. Good GPU. Okay, could you please mention a few free software for deep learning and how we can contact you for further future collaboration? Okay, for Python, you can use Jupyter. Okay, Jupyter, and I think it's free. Yeah, Jupyter free. So you can use PyTorch. Okay, Keras.
PyCharm also.
Yeah, PyCharm. Sorry, Keras. TensorFlow. There are a lot of sources. Okay, you can email me Okay, Azamimi, Unimed.
You write down here.
Edu that.
Yeah. Just
Yeah,
write
yeah.
down.
I think you have my email.
Yes, yes.
Yeah. Yeah. So for this kind of images, because if you deal with images, there are very big data. So like you have to. It's better if you can have a GPU, okay, instead of just a cloud notebooks. If you use Google Colab, you have to pay for the extra memory. Yes. Okay, any other questions? Okay, this one running. Okay, now its results come out already. Okay, like if we use this Kaggle notebook, I think for beginner, you can use this notebook to learn about the coding. Okay, I also ask my student to learn using this so that they know this coding. Okay, and the output is what. So for example, this one the file, and okay, this is the image data. Okay, so 1 is for the methylated, 0 is for unmethylated. 1 is methylated. So this is the sample of images and training stage. This one using Torch, PyTorch, and CNN, training CNN. Okay. Okay, now the result come out. Okay, so this learning rate. So if we use smaller learning rate, so the loss will be smaller. But if we use larger learning rate, the loss will be higher. So learning rate maybe smaller is better. So this is the parameter for the deep learning. Okay, so this is the results. Okay, training loss, validation loss, accuracy, and time. So the accuracy is not good. I think just 0.53. Maybe. Okay, so this one is the results. So for example, the predicted one is means methylated, and the actual is 1. This image, the predicted is 1, but the actual is 0, mean unmethylated. So this is negative. Okay. Okay, so this is the prediction actual loss probability, means for prediction is 1, okay, but the actual is 0. So this is wrong. Wrong prediction. Okay, misdiagnosis. The loss is 1.44 and probability is 0.76. Okay, so this one is the predicted is 0, but the actual is 1. So also wrong. I think many wrong prediction. Many wrong prediction. Okay, and this is the prediction status. So if we give the testing data set, and this is the probability, means if 0.33 means it closer to the 0, means it's unmethylated. If the value is more than 0.59, so maybe 0.6, so closer to 1, means the image more to methylated type. Okay, so this is the testing data, the prediction value, okay, the probability value, and this is the MGMT value. For example, for this is the training data set, so 00114, the ID, the image ID. So it gives the prediction value, probability value is 0.339086. So it's closer to 0, means unmethylated. But for number 4, 00699, okay, the probability is 0.603962, is close to 1, means the status of this image ID, this MRI image is closer to 1, means methylated status. Okay, so still working lah. So you can use this. Okay, just type kaggle.com. Okay, you can find a lot of things there and you can explore more on your own. Okay, so any other questions? Okay, if you have any question or interested to collaborate or further your postgraduate study, you can email me to azamimi@unimed.edu.my.
Anyone having any questions? Just post it. Or you can unmute yourself and ask here.
Yeah. So is
Thank you very much.
yeah, in Malaysia is 6:50 p.m. So late evening.
Okay. Thank you,
Okay.
ma'am, Dr. Azian.
Uh-huh.
And with a
Yeah.
short notice, you have received our invitation and
Yeah.
give you a target.
Hopefully this workshop, yeah.
It will be helpful for our students and participants also. And definitely
Okay.
they're going to collaborate. I'm going to take one feedback form. So whoever will be interested to work in this domain, especially on the brain tumor or the biomedical imaging, definitely through me or directly they can contact you. Thank you, ma'am.
Can we take a photo? Just
Yes,
for
you
my
can.
yeah.
I think you can go to the first page.
Which one?
Or you can stop this one. Stop sharing.
Okay, stop share. So okay, stop
Relax.
share.
Okay. And
Okay,
you can on your video if you can, so that Dr. Aziana can take.
Yeah, can you turn on your camera?
Yes, I will send you. Yeah, students are on their video.
Yeah, this one.
Thank you, ma'am. I've already taken. I'll share you the photo. Thank you,
Okay.
Dr. Paula Bara. Thank you, Azian ma'am. Dr. Paula Bara, I think we'll again connect at 5 o'clock. Dr. Paula? Hello?
Hello?
After 30
No.
after 30 minutes. After 30 minutes again we'll connect.
Okay, thank you. We can.
Okay, we can join again at 5 o'clock after 30 minutes. After this,
Okay.
30 minutes.
See you later.
Okay. Thank you very much.
Okay, thank you.
Thank you.